it is time to learn how to make sperm. That is the process of spermatogenesis. Spermatogenesis is going to happen in the testes. Each testis is covered by a fibrous capsule of connective tissue. That capsule is going to extend into the testis and create several septa. Septa is just the plural of septum. You remember this septum is a division, a dividing structure. And that's going to divide the testis into these lobules. And you can see each lobule is filled with these little tubes. The tubes are called seminiferous tubules. Seminiferous meaning they're going to make the components of semen, and in fact they only make the sperm. The tubules are surrounded by interstitial spaces, and the cells in those interstitial spaces are the ones that actually secrete the androgen hormones, including testosterone. Within the tubules, what happens is the actual creation of sperm, and those are going to be made through the process of meiosis. So this is a cross-section through one of the seminiferous tubules. So imagine if we took one of these little tubes and cut it crossways like this, put it under a microscope slide, this is what we would see. Around the edge, we have special cells called spermatogonia. Those spermatogonia are going to do mitosis and create cells called primary spermatocytes. These are diploid cells. Okay, so they are exact copies of the spermatogonium. That spermatogonium is a type of a stem cell, except it just makes spermatocytes. Here's the interstitial cells out here that are producing the hormones, okay, in the interstitial space around the tubule. So the spermatogonium did mitosis, creates copies of itself. Those are primary spermatocytes. Those spermatocytes are then going to go through meiosis. The first division of meiosis, meiosis 1, they're going to divide into two, separate the chromosomes, the pairs of chromosomes, creating secondary spermatocytes, two secondary spermatocytes. Then they go through the second meiosis division, split again, separating the duplicated DNA in each chromosome, and you end up with 4 spermatids, which are each haploid. That means they have one of each chromosome, and so they have a total of 23 chromosomes. So in the male, in the human male, meiosis happens normally. You start with 46 chromosomes, one cell with 46 chromosomes, you end up with 4 cells with 23 chromosomes. This primary spermatocyte is going to go through this process too. So if we start with 2 spermatocytes, you end up with 8 spermatids. It's just the human female that has to be all extra. Start with 1 oocyte, end up with 1 egg and 2 polar bodies. Okay, so that is definitely a distinction you need to know. As the spermatids start to be released into the lumen of the tubule, they make morphological changes. Morpho just means shape. So morphological changes are just changes in their shape, and they start to take on the shape of sperm. So they start out as these, sort of normal round cells with a big nucleus, an acrosome cap, which is full of enzymes. They have lots of mitochondria, which they're going to need. And then when they start out, they have the normal component of the cytoplasm. As they develop into sperm, the cytoplasm stretches around the nucleus. They build a flagellum. The acrosome cap develops and gets bigger. And they get rid of a lot of the normal cytoplasm because this sperm is going to have one job to do. That is, it needs to swim to get to the egg and deposit its nucleus inside the egg. And so the only things that are really left are the tail to swim with, mitochondria to power the tail, the nucleus, and the acrosome cap, which helps the sperm get through the jelly layer around the egg. So the sperm, once they are done, once they're, excuse me, spermatids, starting to take on that final sperm shape, they leave the seminiferous tubule and they enter this structure called the epididymis. Now, this is one problematic term, epididymis, because it's so hard to spell, but you do need to know how to spell it. So I learned that the only way I could spell epididymis correctly is to say Epididymis. So that's how I say it when I have to spell it. It starts with an E, and then everything else is an I except for this Y in the middle. So Epididymis. Epididymis. In the epididymis, the spermatids develop into this final sperm shape. The acrosome cap is full of enzymes that are going to be able to penetrate and eat their way through the jelly coat, which is the part of the follicle that left the ovary with the egg. Then the nucleus makes up the rest of the head. This is surrounded by plasma membrane. There's a little bit of other cytoplasmic stuff in here, but most of it's gone. Then we have a midpiece that is jam-packed with mitochondria. Those mitochondria are going to produce the ATP to power this tail so that the sperm can swim. Hey everybody, here's what's important. Sperm swim. Let me just say that again. Sperm swim. Sperm are tiny. Sperm are so very tiny. I cannot impress upon you enough how tiny sperm are. They can swim through your clothes if those clothes are wet. The holes in the fabric of your clothes are like just giant portals to a sperm, and sperm swim. Just remember, nature has one job to do, and that is to make more of nature. And it's really good at it. Use a condom. We'll stop there, and then we'll learn about the rest of the reproductive system and what happens to the sperm after they leave the epididymis.